So thanks for coming today. And what I thought I'd do is we're starting a new series, seminar series, to tell you about some of the exciting science going on at Scripps, Florida, in Jupiter. What I thought I'd do to kick off this series is actually kind of overview everything going on at Scripps, because it's really one institution with sites on both coasts. So let me tell you what our vision is for Scripps and where we're headed. When I took over the leadership of Scripps, Scripps, as Doug said, is an outstanding scientific institution, but I think we wanted to ask whether we could do something even more and create a new model for nonprofit research institutes that could have a very significant impact on science and how we do it. So we're creating a new vision, and that vision is not only to advance scientific discovery, which we've had a rich history of doing, but accelerate the impact of those discoveries on public health. So how do we increase the rate at which we go from understanding, human physiology and disease processes to making new drugs for unmet needs like cancer, degenerative diseases of aging, and childhood diseases? And importantly, if we can do that, not only can we accelerate the impact of science on the public, but we can actually create a new model for sustaining nonprofit research institutes, because by partnering the new medicines we create with pharma, we get revenues back that now we can reinvest in the research enterprise and grow the research enterprise in a self-sustaining way. So those are our goals, not only to do great science, make new medicines, and create a new model for funding nonprofit research. As Doug said, Scripps has two sites. We have about $300 million a year in funding of our research. And we have roughly 200 labs, 2,000 coworkers, and roughly in Florida, 50 faculty. As Doug also mentioned, our focus is in chemistry and molecules, but also in neuroscience, structural biology, computation, and immunology, which is a field that Scripps was actually founded on. We have a great graduate program with graduate students in La Jolla and in Jupiter, a great faculty, and as Doug said, we were actually ranked number one in the world for our impact on innovation and science, higher than Caltech and MIT, okay, which was pretty impressive. A little surprising to us, but impressive. So that's Scripps, and as was also mentioned, we have a rich history of impacting not only science, but also technologies. We've created new technologies for drug discovery, from RNA-based drugs that target RNA to large screening libraries and screening systems. We have probably the largest effort in rational vaccine design. We're really well known for developing new synthetic methodologies to make molecules easier, and genomics and proteomics technologies to understand the human genome. But we also have made many advances in basic science and sensory neuroscience. How do you feel pain? How do you touch? How do you taste? And in understanding the immune system, the innate immune system, and how the immune system recognizes foreign molecules and discriminates those from self at a molecular basis. So that's kind of the history of the Institute. And so what you realize is not only here, but there are a huge number of exciting advances being made around not only the United States, but the whole world. In fact, it's the golden age of science. We understand more about the world around us, including our own bodies, than ever before in science. But somewhat surprisingly, we're still making drugs for human disease at roughly the same rate that we have in the past. So what is the mismatch here? We understand far more than we ever have in the past, but it's taking us just as long to make new medicines. And clearly there's a huge need for huge medicines in cancer, aging, childhood diseases, neglected diseases, and so forth. And so why is that? And you could argue it's a result of the fact that most scientific discoveries are made in academic institutions, like Scripps, like Rockefeller, like MIT, Harvard, and others. But most of those institutions are ill-equipped to turn those discoveries into drugs. We don't have the expertise, the infrastructure, or the processes. So what happens is that knowledge transitions to big pharma and biotech. They have the capability to do that, but they have a very different culture, okay? Different focus on science, on commercialization, on intellectual property, and even different cultures. So that really creates an ineffective coupling that slows the whole process. And so we asked whether we could actually begin to go all the way from the research bench to a patient's bedside, all under one roof. The Scripps roof is a nonprofit institute. So how did we do that? Well, many people have tried to build drug discovery capabilities in academic institutions, and the difference in cultures and what you need to do that has created fairly large barriers. And so those attempts haven't been all that successful. What I did, as Doug mentioned, when I moved my lab to Scripps, I also built a genomics drug discovery institute with the Novartis Research Foundation, and really we learned how to discover drugs, build technologies to do it faster, cheaper, apply those technologies, and had a real-time learning experience. Novartis, over the 10 years I was running the institute, gave us about $1.5 billion. So it was a really great education, okay? And luckily, we made a lot of mistakes along the way. Not luckily, we made a lot of mistakes along the way, and luckily, Novartis gave us enough funding that we could just pave over our failures with their money, okay? But the take-home lesson is it taught us in real time how to do this. And so we asked, when I came back to Scripps, a few years later, we asked whether we could do the same thing that we did with Novartis, but in the nonprofit setting with graduate students and postdocs and faculty. And so that was the experiment we ran. It was closely affiliated with Scripps. It was called Caliber, and it's since been funded by a large number of organizations. Our largest partner is the Bill and Melinda Gates Foundation, which has given us roughly $130 million. But we have collaborations with other foundations, with pharma, and we've built a lot of the capabilities and expertise you see in large biotech and large pharma companies within a small organization. Part of the way we did that was not by doing everything ourselves, but also by collaborating with the rest of the world. So we know what we do well, we know the biology others do well, not only at Scripps, but at institutions throughout the world. So we form partnerships with many, many different labs to advance discoveries to medicines. So this is just kind of an overview of the infrastructure we have. We make proteins as therapeutics, we make small molecule therapeutics, we have large chemistry efforts, pharmacology, safety, we have regulatory people, we carry out IND enabling studies, and now we actually submit INDs, get FDA approval, and put drugs into patients that we made with other clinical partners. As I said, the model is highly collaborative. So if somebody has really neat biology, for instance, to treat tuberculosis at Cornell, we partner with them, work closely with them, and try and make a new TB drug. So, given we set up Caliber as a drug discovery focused institute, and we have Scripps, who's an outstanding basic research institute with interest in translational research. We said the easiest way to build this bench to bedside model is just to combine the two. And at the time, I happened to be president of both institutions, okay? So it was pretty easy to do, okay? So in 2018, Caliber became formally the drug discovery division of Scripps Research, okay? So the idea is now you combine a terrific basic research institution and capability with a lot of very bright faculty, grad students, postdocs, great federal funding, very innovative, with the capability to turn those discoveries into drugs with the expertise and infrastructure to make drugs. And that makes the whole process really one smooth, integrated effort. So that was the idea. So we did it. Question is, everybody wants to do this. Is it working? Okay. It's simple to have a vision. It's somewhat hard to make it a reality. The first thing we did is rebuild the Scripps board, okay, with people who were very interested in what we were trying to do and the model we were creating. For instance, Gerald Chan, is the Chan School of Public Health at Harvard. He's incredibly interested in what we're doing. John Martin was the pioneer that built Gilead and was the chairman of the board of Gilead, so probably one of the best drug discovery people in the world. Will Hurst of the Hurst Foundation is very interested in kind of bridging basic and applied research. So we've built a really supportive group of people to help us get this done. And so where are we? What's the scorecard in terms of what have we accomplished? So this is an example of one program we've created. It's called the Primer Program. This was a program where we created a number of new drug candidates. And the question was, how could we actually move those into the clinic? Because it's pretty expensive. So we created a program where people would give recoverable grants or gifts to help us put these molecules into the clinic, make new medicines. If anything in this portfolio was successful and we partnered it for further development with pharma, we give the donors their money back with the only constraint that they have to use it for a charitable purpose, okay? So that was the first mechanism we created, and the first donor to Primer was Bill Gates, okay, because he's really excited about the model. So what is in that portfolio? Well, right now we have 3 clinical stage programs, 2 for cancer and 1 for osteoarthritis. Over the next 2 to 3 years, we think we'll put 4 to 5 additional molecules into clinical trials. We think we'll file roughly 2 INDs a year. And behind that, we're creating a robust pipeline. So the fact that an academic institution has put 1 drug into a phase 1 study is probably unprecedented. Doing 3 and 2 in 1 year, I think it's really starting to validate the fact that this can be done in the nonprofit sector. So as a couple of examples of programs, this is a program that started from a discovery at Scripps and went all the way through a small molecule drug and now is in a 60-patient phase 1 clinical trial for osteoarthritis. The only way you treat osteoarthritis now is you take painkillers and sooner or later you take your knee out, okay, and have it replaced. So a better approach is actually to repair the damage in the disease. So what the approach was, there's a certain class of stem cells in all of our knees. They're called endogenous adult stem cells. And we found a molecule that turns the stem cells in your knee into chondrocytes, which are the cells that make cartilage. So you get a little injection in your knee, you make chondrocytes, the chondrocytes deposit down cartilage, you repair the damage, okay? So that's right now in patients. We've taken a similar approach to multiple sclerosis. It's an autoimmune disease that demyelinates the axons, which are the wires that connect the neurons in your brain. We've actually found a molecule that will form the cells in your brain that make new myelin. So we put myelin, the insulation, back on the wire, and we reverse the disease. So that's going into a phase 2b study the end of this year, early next year. We also have molecules that make new lung for people with COPD and lung fibrosis. And we think we have a therapy that could actually, for people who have had heart failure, you can take this molecule after heart failure and it'll make new heart muscle, okay, which would be a big deal, okay? So one program and approach we have is actually to repair the damage. I really got into this when I turned 50, okay? And the good news is I'm a chemist, so you can do something about it if you work on a young enough age, okay? So that's one program. We also have a program, and we're recruiting patients for in a 60-patient phase 1 trial for metastatic prostate cancer. If you have prostate cancer and it reoccurs and it becomes metastatic, there basically is no treatment for it. So we're developing a clever way, since the cancer is basically drug resistant. We've created molecules, sophisticated molecules from technologies developed at Scripps that grab onto the killer T cells in your body that normally are used to fight infection and bring them to the prostate cancer cells so that they now kill the cancer cells instead of a virus or a pathogen. So this is in preclinical models with tumors that we took from patients that had metastatic prostate cancer. We absolutely ablated it. So we're pretty excited that this might have a big impact on prostate cancer. There's another really exciting therapy developed over the past 5 or 10 years, called CAR T cells. This is an idea that's similar, but here you take T cells, these killer T cells from the immune system out of your body, you genetically reprogram them such that when you put them back in the body, they target cancer. And so these therapies have been developed, and shockingly, in young adults that have failed every other therapy, with 6 pounds of tumor burden, they've actually cleared the disease and it hasn't come back. So it looks like it could be a curative therapy for some cancers. The problem with this therapy is it's a living drug. You take cells, you put them back in the body. Once you put them in the body, you can't control them. If I get a headache, I take 3 Advil. If it's not cleared, I take 4. Sometimes I go to 5, okay? I can dose myself. With this therapy, you can't. So its safety is an issue, and it can't go into solid tumors like breast cancer and others. So we've created a way at Scripps to absolutely control this therapy. So you can titrate it on till it's effective, stop, it's still safe, and then turn it off, okay? And we've also made it, we think, cheaper to implement. So this is right now, an IND was just approved by the FDA, and we're going to start recruiting patients for this trial at 5 different sites in the United States. We're also looking at neurodegenerative diseases. A huge, interesting aspect of neurodegenerative disease now is people are now focusing on inflammation in the brain. And the immune system and the immune response to the damage in the brain is starting to become, people believe, play a major role in many different neurodegenerative diseases, from ALS to Parkinson's to Alzheimer's. And we need new ways to tackle and reduce that inflammation. So we're using another kind of immune cell called a T regulatory cell. And we have interesting ways to make drugs that will increase the number of Tregs in somebody. And recently, people increased the number of Tregs in a small clinical trial with Parkinson's patients, some of whom were immobile, and they started walking. And they actually infused these T cells into ALS patients and actually stopped the progression of the disease. Both of those experiments were complicated in the practical implementation of those therapies in the clinic. So we've actually created a very simple approach that you need a shot once every 4 weeks, we think, and it'll have the same effect. So we're really excited about moving this forward. And I was talking to the CSO of the Alzheimer's ADDF Foundation yesterday at breakfast, and I think they're excited to fund this, as well as Michael J. Fox. And another example is many people suffer from intestinal diseases, and one way you treat those is with anti-inflammatories. But the big problem is the gut lining is leaky. So you have a bunch of bacteria in your gastrointestinal tract that leak molecules into your body and vice versa, and that creates a lot of the inflammatory response. What you want to do is repair your intestinal wall. So there's a natural peptide that's known to do that. The problem is it's very short acting, and so it has a half-life of 20 minutes, and it's not that effective. So we've used, again, Scripps technology to engineer a long-acting form of that, and we're on track to take that into ulcerative colitis, hopefully with a Mayo Clinic in the next 2 years. So those are some examples. So is it working? I think we're putting molecules in the clinic. Another question is, are the pharmaceutical companies and biotech companies realizing the value of what we're doing as an academic institution? So I had a meeting with the head of corporate strategy and the head of R&D at AbbVie, and they got really excited about the fact that an academic institution could actually put drugs in the clinic. They created a partnership with us. The first partnership was to move that CAR T-cell therapy into the clinic, and importantly, they're paying all the costs of the clinical study, even though we own the molecule. So they're enabling us. And then if it's successful, they pay pretty significant milestone and royalty payments to us. So we benefit twice. They actually asked us to run the clinical trial, which is almost unheard of for a pharma company that has kind of a not invented here attitude to ask a nonprofit research institute to run the clinical trial for them, okay? So I think that's another validation of the model we're creating. Since then, we've done another collaboration where now they want us to present a few programs to them every year of our choice. And if they like them, they'll pay all of the cost to develop a molecule and put it into patients through phase 1. And then give us royalties and milestones after that. So all of a sudden, I think we're starting to see that the model we're creating is being validated by major pharmaceutical companies who have a huge experience in making drugs. We have earlier stage programs, lots of earlier stage programs here in Scripps, Florida, and in La Jolla. As I said, we're making a regenerative therapy for heart failure. We're making new drugs for diabetes, obesity, and liver damage. We're trying to make protective drugs that you can take that activate the body's endogenous protective mechanisms against damage. And the idea is these could be neuroprotective or protective for other organs. And we're working with experts in MDS to actually, this is a precancerous condition where as you go from a stem cell to a red blood cell, differentiation stops and it creates a procancerous condition that turns into AML quite often. There's no treatment for this, so we're working very closely with doctors to screen for molecules that impact that. We also, as I said, our largest partnership is with the Bill and Melinda Gates Foundation. We work on diseases of the developing world, malaria, TB, HIV, childhood diarrhea, and other diseases. That's now been expanded to a collaboration with the Wellcome Trust, the British equivalent. And there we're working on other tropical diseases, Chagas, Leishmaniasis, and others. This is the pipeline we've created with the Gates Foundation. 2 molecules are in the clinic already, one for childhood disease, diarrhea, which in the developing world kills a lot of infants. Another for TB, but we have other programs for malaria and tuberculosis and HIV. As I said, this model's collaborative too. In fact, one of our postdocs went over and spent time in a lab in Thailand, okay, because that's where she could access the mosquitoes she had to access, okay. So one of the most exciting things that we hope to put into the clinic at the end of this year or early next year, HIV and TB kill probably more people than any 2 given diseases at a global level. We've actually made a drug that we think you take like a little flu shot. If you get a flu shot, it's half a mil. You take the same thing. It's not a flu shot, but you take a molecule we made every 6 to 9 months, and it protects you from HIV. So it's a chemical vaccine, in essence, that will stop transmission of HIV, which people have been looking for since HIV was discovered, okay? So we're working closely with the Gates folks to develop this for as a drug for the developing world, which will be given away at cost, okay? But we're also now working with a big pharma partner to have this as a drug for the developed world too, so it has a global impact. So we're really excited about that. Another thing we've done with the Gates Foundation is there's a realization that many of the known drugs we take will impact more than one disease, okay? So thalidomide was created as a drug for disease X. It turns out it ushered in the best therapies for multiple myeloma. AZT was made for cancer. It was the first HIV drug. So this is called drug repurposing, and the idea is many drugs we take will work in other disease settings. We just don't know it. So with the Gates Foundation, we made the largest collection of every known drug that's been put into people or approved or late-stage preclinical. It's about 14,000 compounds. And so what can you do with this? Well, as I said, we were working on childhood diarrhea, so we had a postdoc start working on this with funding from the Gates Foundation, set up a screen using cryptosporidial infected cells. And from the day that postdoc started the program till the time we were treating kids with the drug, people with the drug, it's 2.5 years. Turns out we found an old Novartis leprosy drug that kills cryptosporidium better than anything anybody's ever seen. So we've now found repurposed drugs that make new lung, for example. So Gerald Chan, who's a big proponent of public health and a huge philanthropist in the area, is partnering with us to actually create a drug repurposing initiative. And the idea is if we can find known drugs that work for new diseases like fibrosis or Lyme disease or what have you, you can develop that drug faster and a lot cheaper, okay? So it's really to address not only the speed of making new drugs, but the increasing burden of healthcare financially. So 2 examples is we found a known drug used to treat infectious disease that we think will reverse scarring and keloids. So we entered into a collaboration with Cincinnati Children's Hospital, which is one of the top 3 hospitals in the United States, and we're actually going to initiate a clinical trial with kids who have severe scars or keloids to reverse those with a drug that's right now used to treat infectious disease. Another example of repurposing is we were interested with the Gates folks in stopping the spread of malaria. So the idea was one way to block transmission that's been modeled is you take a drug, and every time a mosquito bites you and takes a blood meal, it draws some of the blood and it dies before it infects somebody else, okay? So that's been modeled to if you impact 50% of the population in a village taking that drug, you'll stop transmission of malaria 90%. So the way we got that drug was we were looking for things that killed mosquitoes. So I wrote to the head of DuPont, who was really terrific. This is an ad for DuPont, and gave us all of their insecticide collection to screen against malarial mosquitoes, Anopheles. And we found a drug that worked, and it's related to the drug. If you have a dog like me, you give a dog flea and tick, basically the same drug. So we're developing this with the Gates Foundation for people. You take dog flea and tick once a month, and you don't spread malaria, okay? But every now and then I go back into my lab and do science and talk to the scientists, and I'm like, guys, dog flea and tick. You give it to your dog, kills mosquitoes, it kills sand fleas, kills ticks. All these dogs running around in the northeast, just, in fields laden with Lyme-infected ticks. Do dogs get Lyme disease? The answer is, of course they get Lyme disease. Well, how come all our dogs don't have Lyme disease and we do? It's because they're taking dog, flea, and tick, okay? So we're now, as part of this repurposing initiative, going to turn that into a drug, go through safety studies. It's a very safe drug. And the idea is in the beginning of the summer, you take a pill. You don't care if a tick bites you. It dies before it transmits Lyme disease to you. So it will absolutely, we think, block transmission of Lyme disease. So that's another example of repurposing that we're doing, and we're doing it with the terrific support of Gerald, who's a wonderful human being and a great philanthropist. We also are starting to look at childhood diseases. Two of the efforts we're focused on right now, there's an effort in Jupiter around understanding SYNGAP1, which is what's called a rasopathy, as well as Noonan's disease in Cincinnati. These are two childhood diseases that affect 1 in every 2 to 4,000 kids. It's pretty frequent. Noonan's disease, 20% of the kids die of heart failure. And SYNGAP1, the kids get epilepsy and have other disabilities. And so this disease is caused by mutations that turn on a signaling pathway in cells that people have targeted with anti-cancer drugs, because that pathway is also turned on in a number of cancers. So drug companies have made drugs to target that pathway to kill cancer cells. And the realization that people had in Cincinnati, and we're working with them, is that you can turn off that pathway with a cancer drug. Turns off the pathway. Kids get all kinds of bad side effects from the cancer drug. Can't take it chronically. Whoa, you don't have to kill the cells, you just have to tune them down a little. Drop the dose, factor of 10. Kids' cardiomyopathy disappears. All of the side effects of the drug disappear, okay? So this is another case where now we're going to a major pharmaceutical company. We've asked them for their MEK inhibitor. We think they're going to give it to us. And it could have a big impact on these childhood diseases. We're also using a drug that we've shown, it's again a regenerative medicine. We've shown it preclinically makes new skin. So there are about 500,000 burn patients a year. And the most important thing you can do is to heal faster to prevent infections. So we're looking at making this as a topical that you can give to burn patients and potentially collaborate with Shriners. It may also work for diabetic wound healing too. The American Cancer Society now, which is the largest foundation that funds cancer research, has come to us and said, we've been funding basic research, patient advocacy, patient support, but we should really be doing what you're doing with the Gates Foundation and funding you to engage the entire academic community to identify new opportunities to make new cancer drugs, turn those into clinical candidates, and put those into patients. So we're actually now working with the American Cancer Society to create a new fund that would be focused entirely on mining the academic community and all the ideas for new approaches to treat cancer. And finally, we kind of have our hands full. So we have a board of overseers in addition to our board of directors, and they're mostly pharma executives and investors in biotech and biotech CEOs. And you guys, this is getting ridiculous. Why don't you take some of these things and actually spin them out as a startup company and hire another group of management personnel to do it and kind of begin to leverage your efforts. So we're right now forming, in the process of forming a new company that Scripps will own half of, okay? And so any returns from that company will go back into the Scripps endowment. But it's to actually tackle all of the damage associated with osteoarthritis, which is to make new cartilage, to repair what's called the subchondral bone, and to repair meniscal tears, okay, which 40% of the time lead to OA. We're also, within that company, putting a new novel therapy for arthrophytosis, which is frozen shoulder, frozen joint. That people at Brigham and Women's realize that if women have frozen shoulder and they get pregnant, they get all the mobility back. So they identified the mechanism of that, and we're working with them to make a drug that you can take once a month to relieve the frozen shoulder. And finally, we also have an effort with Eric Topol, who wrote a book some of you may have read on machine learning in medicine. Had set up an institute at Scripps, and it's very now much focused on precision medicine. So if I get cancer, how does my cancer differ from your cancer, okay? So understanding disease at the level of individuals. He's gotten the largest NIH grant ever given, $250 million to do this. So his group now is part of Scripps too. He decided that it'd be great to join forces with us. So now we go all the way from research to new medicines to artificial intelligence trying to understand disease. So that's exciting. So in conclusion, the question is, can we create a new model for research institutes that really bridges the gap between, historical academic activities like basic research, that bridges the gap between that and making a medicine and actually putting that in a medicine and seeing the impact on a patient, okay? So we've shown we can bridge that gap. I was having a discussion with Bill Gates, and he's like, Pete, normally he's like, tell me what you're doing in neglected diseases of the developing world. So I tell him. And then he said, tell me all the other things you're doing. So I told him. And he said, interesting question is, can you scale this? Because if you can begin to scale what you're doing, I mean, Scripps is big, but it's still small compared to the rest of all of the other institutes in the United States, research institutes and universities in the United States. So I think that's the question we're now being challenged with, is can we actually take what we're doing and beginning to actually scale the impact, to impact a broader range of diseases and even faster. So, and as I said, we do this in a highly collaborative model. Importantly now, some of the molecules we're making that we partner for later stage clinical development are starting to give returns to Scripps that we can then reinvest in the whole research enterprise and begin to make this whole model self-sustaining. So that's what we're doing. That's a progress report. Other folks in this seminar series will follow up with, stories around specific opportunities and give you a little more depth on those stories. And I encourage you to come to those lectures because folks who give them are really terrific scientists who are having a big impact, but also pretty entertaining speakers, okay? Finally, I'd like to thank you for your attention. Yeah.
I had 3 quick questions. What do you?
I'm old enough I can only remember one at a time, okay?
Are you familiar with the Immortal Jellyfish?
No.
Oh, it doesn't die from aging.
I got it.
So what's your impression of how long it'll take to cure aging with quantum computing? If quantum computing's 100,000 orders of magnitude faster than the current fastest computer?
So that's an interesting question, is can you extend aging, okay? And it's pretty easy to make a molecule that makes a worm live longer, okay? The question is, why does a worm live so short, is the way you should be asking that question, okay? So people are saying, can we make drugs that help people live longer? Well, first of all, how do you test it? The cost of testing that drug means we're all going to start taking it at 60 and take it till we're, God forbid it works, 120. So it's a 60-year clinical trial that just would be an ungodly cost, okay? I think that the more thoughtful approach is how do you increase the quality of aging, the aging population, the quality of life. And there our focus is on regenerative approaches to reverse disease processes. So if you can actually reverse osteoarthritis or reverse degenerative diseases like MS, okay, these people aren't living in pain or losing function as they get older. So that's the focus that we have, is looking, and also in cancer, I think people are starting to now ask whether you can actually get curative therapies, okay, that absolutely kill the cancer, okay, and stop the cancer. That's our focus for, the aging population. It's not, hopefully if you cure these diseases, you will live longer, but it's important the quality of life you have is really good.
Okay, and is it possible you can study? I understand you spoke with Bill Gates.
Yeah.
He recommended a book called Superintelligence. Did you read that?
No.
Okay, it's a very interesting book. It might be valuable for you to read that and to study the Immortal Jellyfish.
That's a good point. Yeah. Yeah.
I probably don't even need a microphone. My 45-year-old son has type 1 diabetes that he's had since the age of 10. And the insulins have become better. But if you were to find a way to get into his pancreas and to make it work better, you would put all the manufacturers of insulin.
That's a very good point. So we were approached by JDRF, the Juvenile Diabetes Research Foundation. And what they wanted us to do is make a drug that would expand beta cells. So in type 1 diabetics, okay, if you can actually stop the continued damage and you can actually make a drug that expands beta cells and give them back their pancreas, so that's the idea. So we entered into a collaboration with them, and we actually have 2 interesting molecules that if you kill the beta cells in a mouse and give this drug, they remake the beta cells, okay? The question is what's the best way to test these molecules? In type 1 diabetes, you have to give this drug to young kids or young adults. We think it will also work for making regenerating liver. So we've been talking to the Mayo folks, and there are a lot of people who have damaged livers that need to get a liver transplant. The problem is they can't get a liver in time and they die. So the idea is if you could actually expand their liver, you could extend their life long enough that they would be eligible for a liver transplant. So those kinds of drugs are easier to test in that setting than in type 1 diabetics, but we're working, that is the problem we're working on with JDRF. Yeah.
So how broad, in terms of the number of therapies that you're looking at, can you be where you still will have enough specialization that you can make a real impact?
Yeah, that's another good question.
How many?
Right. How broad of an impact can we have and still understand the complexities of a given disease is the question, I think, okay? And I get criticized for two things. I'm not patient, and two, I'm not focused, okay? So exactly what you said. Pete, what are you trying to do? You're working on cancer, you're working on autoimmune disease? So,
It's an impressive spectrum.
So when we started, when I did this experiment with Novartis and formed this institute called GNF, which was funded by the Novartis Research Institute, it was genomics to begin with. And after 3 years, it was supposed to be funded for 10 years. We spent all the money that it was supposed to fund it over 10 years in 3 years. So the chairman of the board, who's now the chairman of the board of Novartis, Jürgen Reinhardt, who's a really good guy and a good friend of mine, looked at me and he said, Pete, you think with all this brain power you have and all these tools you have, you can make a drug? So he pushed the button, okay, you know? And I said, of course, if you guys can make drugs, we can make drugs. So I ran down to the lab and I said, guys, you think we can make a drug? So it turns out the way we started doing that is we interacted with all of the disease areas within Novartis. We partnered with the cancer and made 4 cancer drugs. We partnered with the autoimmune group and made autoimmune drugs. We made drugs for malaria. So we actually figured out how to work in all of these areas, and it was by collaborating with people who knew what we didn't. And the idea is we don't have to know everything. We just have to know who does and form a relationship with them. And it's a lot easier for us as a nonprofit. The best person in the United States in lung fibrosis is at Cedars-Sinai. Paul, I got an idea. Can I come up? Sure, Pete. Spends, 4 hours talking to us and partnering with us. If I were pharma, he would have charged $50,000, okay? But he did it for free, okay? So I think as an academic institution and a nonprofit, it's easier to form those partnerships, and that's why we can work on so many different things.
Thank you very much.
Sure.
The information that you had up there, is it available on the web or something?
I haven't looked. I'm not all that IT savvy, so I haven't ever looked at our website. I actually don't do Facebook and I don't Twitter either, okay? But I think it is. But also, if you want information, the people here like Tara and others are happy to connect you with the right people. So, you can either approach the staff here directly or you can look on our website. And then all of the faculty love to tell people about what they're doing, okay? So the faculty, you can engage faculty directly too. They enjoy it.
You have a wonderful research institute here, and there's another wonderful research institute across the street. Do you compete or collaborate? And what goes on between the two institutes?
Yeah, I think in general, our attitude, not only towards, you're talking about the Max Planck, is that any time we can work together to the mutual benefit of both groups, we do. I think nowadays science is complicated enough. You know, when I started, the idea was I'm going to work on this problem by myself because I know what to do. Nowadays, the problems are getting so complex that the idea that you can solve a problem yourself without collaboration is pretty challenged. So I think there's a real spirit of collaboration and cooperation. Yeah.
You described some incredibly exciting work going on in T-cell research for curative approaches to cancer. Do you think it'll ever be possible where you'll target preventative work to try to develop vaccines that target the development of cancer cells themselves?
Yeah, people are working right now on cancer vaccines. We're actually working on a program that I think will have a development candidate at the end of the year, a molecule that activates your immune system in general to attack cancer. So the idea is if you do get cancer, any kind of cancer, you take an oral pill and it turns on your hyperactivates your immune system, and your immune system now also attacks the cancer cells because as they lyse and they die, they release antigens. And so you may or may not know it, but the way they treat bladder cancer is they give you BCG, which is a kind of mycobacteria. That's the adjuvant they used to use in vaccines. So one of the best therapies for bladder cancer, which is to give BCG, is turning on your immune system to fight bladder cancer, okay? But now people want to be able to do that with a pill that turns on your global immune system. So yeah, I think that there's a huge number of exciting things and it's going on all over the world, okay, not just what we're doing.
We have time for one more question.
Yeah.
The pill you're talking about now, and it works for autoimmune disease, cancer, will it also help for other problems that are caused by your autoimmune disease not working properly?
Yeah, no, the diseases, you can use the immune system to historically fight pathogens, okay, infectious agents. It's now being harnessed to fight cancer. The flip side is if your immune system starts to recognize your own body, you get autoimmune diseases, okay? So the key question is how do you turn the immune system off? And right now it's with drugs that basically turn off all immunity, okay, which create a risk for infection, okay? Can you target those to specific organs so you can protect specific organs? People are trying to do that in type 1 diabetes and, activate the immune system as a protective for type 1 diabetes versus damaging. But each autoimmune disease is a bit different and you need to take a different approach. But there are a lot of neat advances occurring there that over the next 5 to 10 years I think are really going to impact patients. Yeah, last question?
Thank you.
Sure.
Now that you're a fully integrated organization from lab to finished product, are you involved in improving regulatory processes? Because in the past, there have been a lot of obstacles to getting these drugs into the clinic and into the trials.
Yeah, so the question is, we actually only go through kind of what's called phase 1, phase 2A, which is showing proof of concept, okay? The problem is then you have to do a large phase 3 study, which is many tens to hundreds of millions of dollars, okay? We don't have the capacity or the resources to do that, so we form partnerships. The complexities of safety and regulations and so forth and so on about testing drugs in various diseases, you hear people complain about it. But if you actually go visit the people at the FDA, I mean, they just want to help. And it's just sometimes if you have a drug that treats, I was talking to the head of research a while back at Merck. And they have a vaccine that roughly could protect 400,000 kids a year in the developing world. 400,000 if they gave the vaccine. That vaccine and testing has a safety issue that affects about 1 in 100,000 kids. Okay? So if you give it to 400,000 kids, 399,996 are okay and saved. 4 may die. You can't develop that drug because it's not approved in the United States because the side effects are too. So that means sometimes we aren't making rational decisions about, what to approve and what not to approve for what indication, and I get why, okay? But I think that's more a societal problem, and as the healthcare costs go up, there are new therapies being developed. Orphan diseases are now, many pharma are working on them. And what they realize, I shouldn't say this, okay, because some of them are our partners, but what they realized is if it's an orphan disease and there are 10 times fewer patients, you can just charge 10 times more for the drug, okay? So there are drugs being developed to save kids that cost $500,000 a year. Does anybody not want to save a kid by using that drug? No, the answer is we do. And that's why Gerald Chan came to us and he said, if we can use repurposed drugs for that, all of a sudden they become a lot more cost effective and lower cost. So these are the kinds of things now we're starting to focus on. So that's the purpose of our whole repurposing initiative, is to see not only can we make a drug, but can we make it affordable to a kid that has to take it from the time they're 5 to 70. Yeah. Thanks a lot.